In this lesson, we learn how the cell cycle is regulated. All right, so if you have a person that's, say, recovering from a broken bone or some kind of a wound, that healing process relies on the cell cycle to make new cells in order to repair those damaged cells in those tissues. So, in the human body, a lot of the cell types don't actually continue dividing. For example, most muscle and nerve cells don't continue dividing once they've developed. But other cell types, like our skin cells or bone marrow, even the cells lining the digestive system, they all grow and divide rapidly and on a very regular basis. So, why do you think certain cell types would need to grow and divide more quickly and more frequently than others do? Well, the reason for that is because cell division is critical for repair to the body as well as for the general maintenance of body tissues. So, if you were to consider how the body's cells know when to divide and when to stop dividing, you might have some questions there. So, we have this girl in the photo here, and you see that she has a cast on her leg, or maybe you didn't notice that, but if you take a look, she has a pink cast on her leg. She's got some crutches behind her, and so she's broken her leg. Now, how do the cells in her broken leg know that they need to divide more quickly than those cells that are in the bone of her healthy leg, the other side? Well, scientists have answered these questions by doing laboratory experiments. So, when you have a cut, what kind of a healing pattern do you see? Where do the new cells form first? Well, the cut heals from the edges and work inward. So if you ever watched your scab, it gets smaller, right? And you begin to see some pink uh, area around that wound on, say, it's on your arm or your hand or something, and that is a sign of fresh new cells. So it heals from the outside, from the edges towards the middle. So the image here on the right of the screen is actually an illustration of how the break in the x-ray that's on the left there would heal. Okay, so this is the bone. Got the bone here, and you can see the break. And of course, in an actual x-ray, it wouldn't be red like that, but they made it red just to make it stand out a little more so it's easier to see. And you can see that illustration of that same break right here and the process of it healing. So we get these new bone cells forming, and they keep forming from the outside towards the inside. So if these cells on the outside, right here, are dividing and replicating quickly, when do you think it becomes likely that they're going to stop dividing so rapidly? Well, it's when they meet up with neighboring cells and they therefore know, because of the stimulus of there being other living cells next to them, they can actually stop dividing. They can slow down that cell cycle. And this is due in part to some stimulus and response mechanisms that are built into all living cells. Okay, there is a sort of a communication pathway for cells to understand, oh, there's a bunch of cells next to me, I'm going to stop. Now, they don't think about it like that. That's how we think, right? We think things through logically, and if this is true, then this is true, and then I can respond in this way. Cells just happen to get a signal that there's something next to it, and that they don't need to continue replicating. It's pretty fascinating, in fact, how it works. It's a very complex system that allows for that to happen. All right. So again, the cells at the edge of an injury are stimulated by the lack of cells next to them. And so they start to go through the cell cycle, dividing much more rapidly than they would otherwise. So as the injury heals, the rate of cell division begins to slow. Now, there are these things called cyclins. And so scientists could describe what's happening as a wound or a bone heals, but they still, before they found these cyclins, they still tried to figure out what exactly was that signal that actually regulates or controls that cell cycle, like I was just talking about. Well, what they needed to do was find what tells the cell when to divide, when to duplicate its chromosomes, or when to enter another stage of the cell cycle. So in the 1980s, this wasn't that long ago. I was in high school in the 1980s, middle school and high school. Scientists at that time found a protein in a cell that was going through mitosis that wasn't present in a non-dividing cell. So when they injected that protein into a non-dividing cell, all of a sudden a mitotic spindle forms. Okay, what is a mitotic spindle, you might ask? Mitotic spindle is just a fancy way of saying the spindle fibers formed out of the centrioles, which you've already learned about. Okay. So, kind of a recap there. Scientists found a protein in a cell that was undergoing mitosis, and it was a protein that they didn't find in non-dividing cells. So they're like, wonder what this cell does. Wonder what this protein does. Why is it in this cell that's dividing, but we don't find it in cells that aren't dividing? So they inject it into a non-dividing cell, and lo and behold, that mitotic spindle starts to form. So they named this protein a cyclin, and there are a number of different cyclins that they've found in the time since. But these proteins are what help regulate that cell cycle. So that's that complicated stimulus and response system that I was telling you about. So the mitotic spindle, to get even a little more in depth on that, it's basically a structure that grabs onto the duplicated chromosomes at the centromeres, and it pulls those chromosomes apart so that those two copies of the chromosome can each get divided into one of the two new daughter cells. So again, they called this protein a cyclin because it seemed to regulate the cell cycle. So it sounds like cycle, right? Cyclin. So that's a kind of an easy way to remember. Oh, what was that protein that regulates the cell cycle? Oh, it's something like cycle. Oh yeah, cyclins. Okay. So there's a little trick for you to remember that. Now, let me ask you this. What could go wrong if the cell cycle isn't fully and carefully regulated? Well, the answer to that is that the DNA might not actually be divided evenly between the two new daughter cells. So those cells couldn't divide when the organism doesn't need new cells. Or let me rephrase that. So these cells could then end up dividing when the organism doesn't actually need new cells because they don't have the right information or all of it. And so this could waste valuable resources, and it could become very problematic in terms of becoming cancerous. All right. So scientists, as I said before, have found a number of other proteins that help regulate the cell cycle as well. The cell cycle is controlled by lots of different regulatory proteins. Some of them are inside the cell, and some of them are outside of the cell. So the internal regulatory proteins respond to events inside of the cell, and they let the cell cycle proceed only when certain steps have already been completed. For example, some regulatory proteins actually function to keep mitosis from starting until the chromosomes have been duplicated. Another regulatory protein keeps the cell from proceeding to anaphase until the spindle fibers have attached to the chromosomes properly. What do you think The role of external regulators is, well, if you're thinking maybe they respond to events that happen outside of the cell, that's an excellent guess. Because that's, in fact, exactly what the external regulators do. The external regulatory proteins direct the cell cycle to either speed up or slow down. And there are growth factors that stimulate the growth and division of cells. So, other external regulators can actually have the opposite effect, causing the cells to even stop or just completely slow down their cell cycles. Stopping or slowing a cell cycle can be just as critical as stimulating the growth. Stopping the cell cycle can actually prevent excessive cell growth and keep body tissues from disrupting one another. All right, so uh, a lot of new cells are constantly being formed in a multicellular organism. But at the same time, there are a lot of cells that are dying. And some will die due to just normal wear and tear, but others are actually programmed to die. And the process of being programmed to die is a process called apoptosis. You see that here on the screen. Now, apoptosis plays a critical role in structuring the body during growth and development. Imagine if old skin cells didn't die and shed. What would happen to a person's skin if that were the case? Well, if you think about it, the skin would keep getting thicker and thicker and thicker, and eventually it would lose some of its properties, like its elasticity and its ability to sense heat and cold and things like that. So this idea of apoptosis, this programmed cell death, it is really a very normal cell process, and it's actually very beneficial to pretty much every organism. In fact, apoptosis is completely necessary. Um, it's a completely necessary process for all multicellular organisms. Now, apoptosis allows organisms to really more fully control which cells continue to grow and divide and which don't. So, if a cell is old or if it's damaged, apoptosis allows this cell to basically be destroyed rather than for that cell to continue growing and dividing and using up all the resources that could be used by other healthy cells, resources that those healthy cells need to survive. All right, so I'm sure most of you are probably familiar with cancer. You probably know somebody or know of somebody that has either survived cancer or maybe even passed away from it. Cancer is one of those things that we understand a lot about it, but we haven't quite figured out how to prevent it. But definitely, there's going to come a day, we hope, that we can actually prevent cancer. Now, some cancers are completely preventable. Skin cancers and things like that are often preventable just by avoiding getting too much sun. Um, some lung cancers are avoidable by not smoking cigarettes or using vapes or using other products that are inhaled, because those can damage the tissues and the cells that make up those tissues inside the lungs, and they can turn to cancer. Now, cancer is really a disease of the cell cycle, and that's why we talk about it in the chapter that deals with the cell cycle. So, there's an area in this image that shows cancer, and that area is this space right here. Okay, that is a tumor that's growing. And so we have a blood vessel down here, and that's just underneath the normal tissue. But then up here is a tumor that's developing, and that is basically a mass of cancer cells. That's all a tumor is a mass of cancer cells. So how do you think a tumor like this forms? Well, the answer to that is that once a cell starts dividing too fast, there's no control over the cell cycle. Um, that begins to cause there to be more and more cells with that same problem. So one cell gets out of whack, it starts dividing, and all of the cells that it divides into, and all of those cells divide into more cells, they all have that same problem. Before you know it, you have a big mass of these rapidly dividing cells. So basically, those cells aren't going to respond to any of those internal or external regulatory signals that we're talking about just a few minutes ago. So why do you think a rapidly growing mass of cells can be harmful to healthy cells? Well, it's important to realize that cells need resources in order to grow and divide, just like we need resources like food and water. In order to survive, our cells need resources as well. When cancer cells are just growing uncontrollably, they don't only displace healthy cells and tissues, but they also use up those resources that normal cells need. Now, they displace healthy cells and tissues by taking up space that those healthy tissues and cells would otherwise take up. Okay, and then they use up a lot of the resources that those normal cells would actually need. So the cell cycle is disrupted, and these cells just start growing and dividing uncontrollably. So here we have a series of images, and they basically show the formation of cancer. So the first step in the process is a cell begins to divide abnormally. Okay. And then in step 2, the cells produce a tumor and start to displace normal cells and tissues. And then in this third step, the cancer can actually start to move to other parts of the body. Now, this is when cancer gets extremely dangerous, because when these abnormal cells break free from their original site, they can now move to other parts of the body. They can get into the bloodstream or the lymphatic system and begin to move to other areas of the body, and then these cells begin forming tumors in these other places as well. So this starts to produce secondary tumors. And so when these cells begin to move around the body and start creating secondary tumors in other places, this process is called metastasis. Now, there are a lot of different triggers for cancer, but but all of these ultimately disrupt the genes that regulate this normal cell growth and division. The result of the gene defects is that some cancer cells no longer respond to the external regulators, and others no longer produce internal regulators. So the defects in these genes can be caused by things like I said before, things like tobacco use, exposure to radiation. That could be radiation from the sun or radiation from nuclear waste. It could be other defective genes in the cell, and it can even be caused by viral infections. So control over the cell cycle is broken down in all cancers. So all cancers are essentially the same. We give different cancers different names due to either where they originate or what caused them. Okay, so cancer basically results from a defect in the genes, and those defects can either be inherited in additional cells, or they can be caused by the infection of a virus or exposure to UV rays or exposure to other chemicals that we refer to as carcinogens, cancer-causing chemicals. So if you have a tumor that hasn't spread yet, it hasn't metastasized, oftentimes they can perform surgery to remove that cancer, and it's usually a pretty effective treatment. This is a very common treatment for skin cancer. Radiation is another way that we go about treating cancer, and that will actually destroy the DNA of the cancerous cells. And since they need that DNA in order to quickly and rapidly divide, that basically shuts them down. They're no longer able to continue duplicating their DNA fast enough to be cancerous. And there's also a therapy called chemotherapy, and this basically kills cancer cells or just slows their growth. So, what unintended effects might radiation and chemotherapy have? Well, if you know anybody who's been through those treatments, you probably know the answer to this. The problem with those particular treatments, they're very effective, but they're very difficult on the patient because they actually can kill healthy cells as well. Okay, and especially healthy cells that are dividing can be disrupted by these therapies. So, the unintended consequences of these cancer treatments are really what's motivating researchers to try to learn more about the proteins that control the cell cycle so that they can find a more specific way to try and target cancer cells and defeat them without harming the healthy cells. Um, Now, of course, um, succumbing to cancer, which is a deadly disease, um, is a worse fate than going through the radiation and chemotherapy treatments. So, at the current time, that's really the best we have right now. And so, many people go through radiation and chemotherapy and they get through it. They might be sick, really sick physically from it, very tired and fatigued and sick to their stomach. They lose their hair all over their body, actually, not just their head, but if they have hairy arms, they'll lose hair on their arms a lot of times, different things like that. But that's all worth it to get rid of that cancer, because once you stop the treatments, then your healthy cells start repairing and replacing all of the cells that were damaged by the treatment. So in the end, it works out usually, not always, but usually. And so it makes it, I guess, worth it. But let's hope that our researchers can find a real cure that can target just the cancerous cells and not the healthy cells around those tumors. Maybe you'll be one of those scientists that figures that out someday. I hope so.